Today we're diving into the microscopic world of bacteria, the tiny organisms that are all around us and even inside us. Bacteria are so small that millions could fit into the tip of a needle. Bacteria exhibit a variety of shapes and sizes, which is one of their most fascinating features. The main shapes of bacteria are cocci. These bacteria are spherical or round. They can exist in a single cell or in clusters like chains, which would be streptococci. There could be in pairs, which would be known as diplococci, or grape-like clusters, which would be known as staphylococci. Bacilli, or the singular form, if there was just one, would be known as bacillus. These bacteria are rod-shaped. They could be short and thick, resembling a pill, or long and thin. Bacilli can also be found alone, in pairs, or in chains. Next, we have spirilla, or singular form would be spirillum. These bacteria have a spiral or corkscrew shape. Next, we have spirochetes. These are long, thin, and flexible bacteria with a tight spiral shape resembling fusilli pasta. They are unique in the sense that they can move in a corkscrew motion. Bacteria are everywhere. They can be found in soil, in water, in air, on plants, animals, and yes, even on and in our bodies. Bacteria have long been used by humans to create food products such as cheese. The cheese making process begins with the addition of starter cultures to milk. These starter cultures are usually strains of lactic acid bacteria such as Lactococcus, Lactobacillus, or Streptococcus. The bacteria ferment lactose, which is the sugar in milk, into what's known as lactic acid. Neutral pH is a pH of 7. Milk has a pH of 6.7. Therefore, the sharper the cheese, the lower the pH. This acidification process causes the milk to curdle, forming what's known as curds and whey. Some cheeses require additional bacteria or even molds to be added later in the process to aid in ripening or aging. For example, Propionibacterium fruitium ricci are used in Swiss cheese to produce carbon dioxide gas, which forms the characteristic holes, and it also contributes to the sweet, nutty flavor. Cheddar cheese uses strains of Lactococcus lactis for acid production and texture development. Yeah, mozzarella also relies on Lactococcus lactis for rapid acid production, which is necessary for the stretchiness of the cheese. There's Parmesan cheese, which utilizes Lactobacillus bulgaricus or Streptococcus thermophilus for a longer aging process, contributing to its granular texture and complex flavor profile. Understanding the role of bacteria in cheese making reveals a fascinating intersection of biology, chemistry, and even gastronomy, where microorganisms are essential to creating a vast array of flavors and textures enjoyed by people all over the world. Now, there's not just cheese. In addition, we have yogurt. The production of yogurt relies on the fermentation of milk by specific strains of bacteria, primarily Lactobacillus bulgaricus. This bacteria is known for its role in the fermentation of milk, where it converts lactose into lactic acid. Production of lactic acid is what causes the milk to thicken and take on the tangy flavor characteristic of yogurt. Streptococcus thermophilus, this is another lactic acid bacterium that works in conjunction with Lactobacillus bulgaricus. These two bacteria are known as probiotics because they can present good health benefits when consumed. But Streptococcus thermophilus typically acts faster than Lactobacillus bulgaricus and is crucial in the initial acidification of the milk. In addition to these primary cultures, some yogurts may include additional probiotic strains like Lactobacillus acidophilus. Probiotics are added for potential health benefits, such as aiding digestion and supporting the immune system. Probiotics also promote good gut health by balancing the good and bad bacteria in the gut to keep your body working the way it should. Some studies suggest the connection between gut health and mood. Therefore, probiotics might play a role in promoting a healthier gut-brain connection. In addition, there is growing evidence that probiotics can benefit the skin, improving conditions like eczema, acne, and even dry skin. Lastly, probiotics have a potential benefits in managing certain chronic diseases, such as heart disease, by reducing blood pressure and cholesterol levels. So these probiotic and bacterial strain cultures also ferments lactose and what's known into lactic acid. The lactic acid lowers the pH of the milk, which causes the milk proteins to denature and coagulate, forming the thick structure of yogurt instead of one that you could pour. Cheese and yogurt serve as an excellent example in teaching about bacteria because it shows how these microorganisms can be beneficial, contributing to both the preservation of food and health benefits when consumed. Looking at the phylogenetic tree of life, the number of bacterial species is immense, and scientists are still working to discover and classify them. Bacteria have been around for billions of years and have diversified into an enormous variety of forms. As of now, there are estimated to be at least tens of thousands of described bacterial species, but the total number of bacterial species existing in nature is believed to be much larger, possibly in the millions or even billions. The vast number is due to the bacteria's ability to live in almost every possible environment on Earth, from hot springs to the deep sea, and from soil to living organisms. Given the microscopic size and the vast number of environments in which they can live, bacteria represent the most numerous form of life on Earth in terms of number of species and sheer quantity. Their diversity is a testament to their evolutionary success. Bacteria are prokaryotic. The term prokaryotic refers to organisms that are classified as prokaryotes. These are unicellular organisms and lack a nucleus or any other membrane-bound organelles within inside their cells. Bacteria are the most well-known group of prokaryotes, and the other group is known as archaea. Let's look at some key characteristics of prokaryotic cells. First, instead of a nucleus, prokaryotic cells contain a nucleoid, a region of the cell where the circular DNA chromosome is located. This DNA is not enclosed by a membrane, instead the genetic material floats freely inside the cell. Pili are thin, rigid fiber structures made of protein that protrude from the cell membrane of bacteria. This pili also is used for adhesion. This adhesion is critical for the establishment of infections in host organisms. Some pili, known as sex pili, play a role in bacterial conjugation. Conjugation is a process where two bacterial cells transfer genetic material between them. Next, we have the flagella or flagellum. Flagella are whip-like appendages that provide bacteria with mobility, allowing them to swim through liquid environments in response to various stimuli. Most prokaryotes have a rigid cell wall that provides structural support and shapes the cell. In bacteria, the cell wall contains a polymer, which is a sugar and amino acids that is not found in the cell walls of archaea or eukaryotic organisms. This molecule forms a mesh-like layer outside the plasma membrane of most bacteria, giving the cell wall its shape and providing protection against environmental stress. The bacteria capsule is composed of polysaccharides, which are long chains of sugar molecules. However, some capsules can be made of what's known as polypeptides, which are long amino acids. This capsule layer serves as a protective barrier, helps in adhesion to the surfaces, and can shield bacteria from the immune response of a host. The immune response of a host is the body's defense system activated to fight off infections and foreign invaders like bacteria. The cell membrane is a thin, flexible barrier that surrounds the cell, controlling the movement of substances in and out of the cell and maintaining its internal environment. Next, we have cytoplasm. Cytoplasm is a jelly-like substance inside of the cell, containing all of the organelles and molecules needed for the cell's life processes. Ribosomes in bacteria are small, circular structures where protein synthesis occurs. Translating genetic information into proteins is essential for the cell's functions. These bacterial ribosomes differ slightly in size and composition than those of eukaryotic cells, making them the target for certain antibiotics. A plasmid is a small circular double-stranded DNA molecule found in bacteria, separate from the chromosomal DNA, often carrying genes beneficial for survival, like antibiotic resistance. As we have seen, not all bacteria are bad. In fact, many are helpful, as we have seen in the production of yogurt's probiotic capabilities and how cheese is made. Some bacteria within our gut helps us digest food. This bacteria play a crucial role in recycling nutrients. Certain bacteria in our gut, known as gut flora or microbiota. These microorganisms assist in digesting food by breaking down complex substances that our own digestive enzymes cannot handle. These also help in processing fibers, starches, and some sugars, contributing to overall digestive health. However, some bacteria can make us sick, like the ones that cause strep throat or food poisoning. That's why it's important to wash your hands and keep food safe. Looking at some pathogenic strains, we will start with Staphylococcus aureus. This bacterium can cause a range of illnesses from skin infection to more serious conditions like pneumonia, meningitis, and toxic shock syndrome. Some strains produce toxins that cause foodborne illnesses also. E. coli. Certain strains are harmful and can cause severe stomach cramps, diarrhea, and vomiting. E. coli is often spread through contaminated food or water, particularly undercooked ground beef and unwashed vegetables. Next, we have Helicobacter pylori. This bacterium infects the stomach lining and can increase the risk of stomach cancer. Next, we have Salmonella. These bacteria are a common cause of food poisoning, leading to symptoms like diarrhea, fever, and abdominal cramps. Salmonella are often found in contaminated foods such as undercooked poultry, eggs, and unpasteurized milk. These pathogenic bacteria can enter the body through various means, including inhalation, ingestion of contaminated food or water, or through breaks in the skin. Once inside, they can evade the immune system, multiply, and produce toxins, leading to illness. In regards to reproduction, bacteria go through what's known as binary fission. Binary fission is a simple and quick process by which bacteria reproduce. It's a form of asexual reproduction, meaning the offspring is an exact copy of the parent. There is a process to this. The bacterial cell copies its DNA. The cell then grows larger and the DNA strands move to opposite ends of the cell. The cell then splits in the middle, creating two identical daughter cells. This process can happen very quickly, allowing bacterial populations to grow rapidly under the right conditions. This can quickly lead to the spread of advantageous traits such as antibiotic resistance throughout a population. In optimal conditions, some common bacteria like E. coli can divide every 20 minutes. This rapid reproduction rate means that a single bacterium can multiply into over 16 million bacteria in just 8 hours under ideal conditions. This rapid and efficient reproduction through binary fission is one reason why bacterial populations can adapt quickly to changing environments, and why bacterial infections can escalate so quickly if not properly managed. The other form of reproduction that bacteria could go through is what's known as bacterial conjugation, and this is a form of sexual reproduction in bacteria. It's a way for bacteria to share genetic information, especially plasmids, which are small circular pieces of DNA. Conjugation helps bacteria adapt and survive in changing environments by sharing beneficial genes. Bacterial conjugation enables the direct transfer of genetic material and is favorable because it encourages genetic diversity, facilitating adaptation to new challenges like exposure to antibiotics in their environment. The good thing is that one could usually treat bacterial infections by taking antibiotics. Bacteria can get nutrition in one of three ways. One, they could be heterotrophic bacteria. There are heterotrophic These bacteria obtain their energy by consuming organic matter. These species can be found in the intestines of humans and animals. E. coli is a heterotrophic bacterium that breaks down and absorbs nutrients from the organic matter in the gut of humans and animals. Most E. coli are harmless and actually are an important part of a healthy human intestinal tract. Next, we have autotrophic bacteria. Autotrophs synthesize their own food from simple substances like carbon dioxide and water. For example, cyanobacteria are photosynthetic bacteria which use sunlight to convert CO2 and H2O into simple sugars like glucose, which also releases oxygen as a byproduct. These organisms are essential for producing oxygen in many aquatic environments. Lastly, there could be chemosynthetic bacteria. Chemosynthetic bacteria obtain energy by oxidizing inorganic molecules such as ammonia, nitrate, or sulfur compounds. For example, sulfolobus are extremophilic bacteria found in hot acidic springs. Archaea is the main group to thrive in these extreme environments. These organisms oxidize sulfur to produce sulfuric acid, deriving energy from this process to fix carbon dioxide. Now, let's look at two species that have the same genus but are different species. Both Streptococcus thermophilus and Streptococcus aureus belong to the same higher classification levels but differ at a species level. So here's a comparison using the classification hierarchy. Both belong to the kingdom Monera, that's where bacteria is placed, indicating that they're both prokaryotic organisms without a nucleus. They are part of the same Firmicutes phylum, which is characterized by bacteria with thick, strong cell walls. Both are classified under the class Bacilli, which includes bacteria that are rod-shaped, and even some that are spherical, which is the cocci. They are in the Lactobacilli A-list order, a group of related bacteria that are often involved in lactic acid fermentation. Both species belong to the Streptococci family, which encompasses a variety of spherical, long-forming bacteria. They are in the same genus, Streptococcus, known for spherical bacteria that often form chains and can be either harmless or pathogenic. Now, here's where they differ in this species. Streptococcus thermophilus is a non-pathogenic species used in dairy fermentation, and Streptococcus aureus is a potentially pathogenic species known for causing various infections. While the classification is almost identical up to the genus level, the significant differences in their behavior, ecological niches, which are the specific role or job an organism has on its environment, and impacts on human health are distinguished at this species level. Going back to E. coli, the diversity in the effects of Escherichia coli strains on humans, ranging from being harmless or commensal, which means beneficial to us, to being pathogenic and harmful, is primarily due to the genetic differences among various strains of this bacterium. All strains of Escherichia coli belong to the same genus, Escherichia, and species, coli, but they differ in terms of genetic variations. These variations are what lead to the different characteristics and behaviors among the strains. In bacteria, even small genetic differences can have significant impacts on their properties. When undergoing conjugation, E. coli strains acquire additional genes. This can lead to the development of new traits, such as toxin production or antibiotic resistance. Furthermore, spontaneous mutations in the bacterial DNA can also lead to new characteristics. These mutations might make certain strains more severe or harmful and allow them to survive in different environments. The pathogenic strains of E. coli, like E. coli O157H7, have specific genes that are not present in non-pathogenic or commensal strains. These genes enable them to cause disease by producing toxins or adhering to intestinal cells. E. coli O157H7 is a particularly notorious strain because it is associated with serious foodborne illnesses. It is known to produce potent toxins and cause severe gastrointestinal symptoms. The O and H are known as antigens, meaning any substance that causes your immune system to produce antibodies against it, and these are crucial for laboratory identification and classification of this strain, which is important for diagnosis and control tracking of outbreaks. Conversely, many E. coli strains in the human gut do not have these pathogenic genes and live symbiotically, continuing or contributing to gut health and other physiological processes. All bacteria are living organisms, and they exhibit all 8 characteristics of life. Let's go through each of them. Bacteria have cellular organization. They're unicellular organisms that are made up of a single cell that contains all the necessary components for life, such as having a cell membrane, ribosomes, cytoplasm, and many more organelles. Bacteria reproduce, either asexually or sexually. Bacteria carry out various metabolic processes, including respiration or fermentation, to produce energy. For example, Lactobacillus species ferment sugars into lactic acid that is used in yogurt production. Bacteria maintain a stable internal environment. The following species, Thermus aquatus, thrives in hot springs, maintaining internal stability despite extreme external temperatures. So we're looking at homeostasis. Next, heredity. Bacteria have DNA as their genetic material, which they pass on during reproduction, and this reproduction could be either binary fission or through conjugation. Bacteria can respond to environmental changes. For instance, that we saw earlier, Helicobacter pylori. It alters its expression of genes in response to the acidic environment of the human stomach. While individual bacterial cells don't develop like multicellular organisms, they do grow in size before dividing. Now, adaptation through evolution. Bacteria can evolve rapidly due to their short generation times. Bacillus anthracis create small bubble-like structures called endospores. Where they are in unfavorable conditions, these can germinate into a bacterial cell again when conditions become favorable. Endospores can withstand drying out, boiling, and many chemicals. Scientists use what's known as an autoclave, and an autoclave is used to create high pressure to completely kill endospores prior to testing. Now, bacteria are remarkable organisms that play essential roles in our world, from supporting our health, to recycling nutrients in ecosystems, to its role in food production. Bacteria reproduce through binary fission, a quick asexual process, and conjugation, a form of sexual reproduction. Their ability to rapidly reproduce and share genetic material allows them to adapt quickly to changing environments, which includes developing resistance to antibiotics. It's important to understand these processes so we can better appreciate the complex dynamics of microbial life and address the challenges of antibiotic resistance. Remember, every tiny bacterium is a survivor, shaped by billions of years of evolution to fit perfectly into its ecological niche. Therefore, you should respect the tiny yet mighty bacteria.